the name?
Yeah, you got it.
With a show of hands, have you ever considered the possibility of microplastics being present within your body right now? Seems like quite a few people have. But for the people who have not had that thought cross their mind, what if I told you that soon enough we could see a significant increase in microplastic accumulation to the point that it could even be passed on to your infant through breast milk? In fact, a 2022 study found tiny plastic particles less than 5 millimeters in diameter in 75% of the 34 breast milk samples that were studied. Microplastic accumulation may pose risks to human health, especially those of infants who are in the critical stages of development, by causing oxidative stress, endocrine disruptors, hyperinduced immune responses, ultimately causing cell death. In response to this alarming issue, we present our product, Booby Trap, an innovative product designed to reduce microplastics in breast milk. Our nipple shield incorporates a molecularly imprinted polymer filter that effectively captures microplastics. It does this by selective interactions utilizing attractive forces that allow the other essential nutrients to pass through, ensuring safer and cleaner breast milk for your infant. Even though it's still in the works, the concept behind molecularly imprinted polymers includes the binding of synthetic receptors to a solid silicone support. It selectively targets microplastics, preventing majority of the microplastics from entering the breast milk. It is a reusable, durable, and heat-resistant product which is easy to clean only using boiling water, making it suitable for everyday life. So whether you're a first-time mom or a mother of 5, incorporating our product into your routine is effortless as it is accessible. In conclusion, by choosing our product, you're not only prioritizing your baby's health and well-being, but also contributing to a future for generations to come. We are currently in the works of receiving expertise from various sources, such as a professor from the pediatrics department at the university and lactation consultants from the Winnipeg Breastfeeding Center. With their feedback and knowledge, we can efficiently cater to our customers and ensure the success of our product. From here, we would like to find a manufacturer that can help develop our product and make it accessible to the public. Additionally, we hope to offer customizable nipple shield designs to cater to different nursing preferences and breast shapes. Thank you.
Thank you, Rabia. Now we'll have some questions from the judges.
Of course.
Thank you so much for starting us off so well. I've been looking forward to this, and you did not disappoint. My question is about your kind of ideal target market, that woman. Have you spoken with any women and gotten any feedback from them?
Yeah, that's a great question. Thank you for bringing that up. Our target audience is parents in general. We have received expertise from a professor at the pediatrics department, and we have also carried out a survey to get in contact with some new mothers from our team members. We were able to speak to them and get a bit of their insight from whether they would use this product or not. We got a few positive responses, but the general consensus was also to be careful when promoting this product to ensure that we are not using or trying to use the fear of parents as well. We want to kind of promote it as like an optional product, as whether they would be careful about microplastics, and that's what we are hoping to do with our product.
So my question's about the reusability of it. So you mentioned that it can be boiled, and so by doing that, does it remove some microplastics? Is there a way, is there specific timing in terms of how long it needs to happen? You know, how many times can it be regenerated? Is this something that can be used for a month or for a year, or?
Thank you for bringing that up. So The idea behind our product is we are hoping it can be a one-time use to make it more accessible and easier to use for parents. The idea would be that it should be washed with boiling water after every use, because if you see over here, the microplastic would be targeted, and with heat, it polymerizes, which then allows it to detach easily. The heat from the boiling water would affect the affinity between the molecular locks and the microplastic template, causing it to detach and ensuring that all the essential nutrients of the breast milk can pass through, but not the microplastics.
Thank you.
This is a wonderful idea, thank you. I've got a question just with regards to the baby latching. Have there been any concerns with that?
Actually, no. We received a lot of positive feedback on the fact that we're using a nipple shield. We initially came up with the idea to use a breast pump, but a nipple shield we just decided was a lot easier, because one thing that we noticed a lot were mothers still want that bond contact with their baby, so we thought it would just be a lot easier. We are hoping to do maybe some small clinical trials in the future to see whether that could be a concern, so thank you for bringing that up, but as of now, it doesn't seem to be much of a barrier.
Thank you, Rabia. Sorry, I wasn't sure if there was one more question. So we're going to hear next from Ano Chimutsa, who will be presenting their project, Planet Zero, a bio-based device for the degradation of polylactic acid plastic. Please join me in welcoming Ano to the stage.
Have you ever been to the Forks and ordered a coffee that came in a cup, just like this? I know I have. Local businesses are switching over to polylactic acid, or PLA, a compostable single-use plastic alternative in response to a growing number of eco-conscious consumers. In fact, in 2022, PLA made up to about 20% of the global plastic production, and this number is expected to grow and even double by 2027. As far as plastic goes, PLA is great. It's made from renewable resources with similar chemical and physical properties to traditional plastics. However, there's a catch. These compostable products aren't as eco-friendly as they may seem. PLA requires sustained high temperatures in order to degrade, which is incompatible with our local composting infrastructure. Our team met with Stephanie Chow, the sustainability technician at the Forks Market, where we learned about the in-vessel composting systems used to process the food and plastic waste produced by the vendors. We learned also that compost has to be reprocessed multiple times, leading to a decrease in overall compost capacity, as well as damaging the blades used to mix the compost. Our meetings with the Canadian Council of Ministers of the Environment further emphasized these issues in similar windrow facilities. In response, we've been working on the design of Planet Zero, a biocatalyst for PLA breakdown, or in simpler terms, plastic-eating bacteria. Our bacteria has been designed to produce PLA-degrading enzymes, which are optimized for these lower temperatures. These enzymes attach themselves to the surface of the bacteria, removing the need for expensive protein purification and allowing for direct contact with PLA. Community members have expressed excitement and joy at our solution because it is easily integratable and easily accessible to local composting infrastructure. This is done by simply adding the bacteria to the compost when it is being ordered, allowing for an increase in PLA breakdown. By diverting PLA from landfills without the need for expensive industrial upgrades. Planet Zero bridges the gap from economic growth to environmental degradation. This aligns perfectly with the sustainability and circular economy that PLA was designed to support. Now, I know none of us want to give up our morning, afternoon, or evening coffee, So let's work together on making compostable plastic actually compostable with Planet Zero. Thank you.
Thank you, Anno. Questions from the judges?
Yeah. I'd love to ask who exactly is the buyer? Like, who is going to buy the product?
We're targeting our products towards people in composting facilities because we already have some compost bacteria. This is similar to that, where they can just add it into their composting facilities and they can use it. So there's no need for any extra training.
My question is, if you were going to move forward with this, what would you do next? What's the next thing?
So at the moment, we have looked at our enzymes and studied our cell surface display system. The next step for us is to transfer our PLA degrading system into eventually a bacteria that is naturally found in compost.
So is this something where, as the product, you would be continuously selling this bacteria to the compost facilities and they would sort of add a batch to each batch of compost? Or is this something where it can be mixed in and will the bacteria grow and divide on their own? Or is this a continual add product? In the absence of PLA, would they die off? Is this something that they need to survive?
Well, technically, it's a little bit of both. So we would sell them at a batch because once the bacteria is in the compost and it's been pasteurized, it kind of dies off. So they do have to get new bacteria for new compost batches.
Okay, thank you.
I have another question. Have you spoken to many composters? Like, I'm interested if there's any reason why they wouldn't do, because it sounds like a wonderful solution. Is there any reason why they wouldn't want to try this product out?
We have spoken to Stephanie Chow. She's the composting person at the Forks Market, and she is interested, and a lot of other people, the Canadian Council, we've spoken to people around the university, and they're all interested in our product, and there's no reason for them to not want it, especially because it reduces the damage and the cost they have to take on to repair the windrow.
Thank you.
Yeah.
Thank you so much.
Thanks, everyone. Our next presenter is Miriam Odatala, who will be presenting their project, Conductive Battery Binder.
So, Canada's zero net emission target requires large-scale changes in energy system, including a transition to electric cars. This transition, however, has one major issue. Batteries are most important and expensive part in the electric cars, but their manufacturing process has significant environmental issue due to the use of toxic material. Simply, this put transition to electric cars, trading one environmental issue with another. The most toxic part of the manufacturing batteries involve battery binder. Binder serves as a glue for connecting battery material together. Unfortunately, the traditional binder relies on toxic solvent mixed with a very harmful forever chemicals and other electrode materials. Furthermore, the use of these binders demands a sustainable amount of energy to dry these solvents and incur high costs for waste disposal. To see how big of a problem this is, I interviewed 61 battery manufacturers who confirmed that solvent toxicity and the upcoming regulation around forever chemicals are their major concerns. Also, these interviews confirmed that the high manufacture cost is a big pain for battery industry. Luckily, we have a solution. To meet these challenges, we developed an environmentally friendly battery binder that does not hurt the battery performance and uses water instead of toxic solvents. Also, while we are trying to associate environmentally friendly solution with increased cost, our solution actually saves battery manufacturers money. With a market size, with a global market size around $5 billion and $1.3 billion in North America alone, we developed our binder. We served our battery manufacturers with a cheaper, more sustainable battery binder that can translate to a cheaper electric cars and for consumers and more sustainable future for everyone. This also leads to provide a cheaper, more sustainable solution, more sustainable batteries for everyone for all electronics in you are using. That's it. Thank you.
Thank you, Miriam. Judges, do you have questions?
So you talked a little bit about cost savings. So your battery binder, how easily is it translatable into the steps to create the batteries already that are available? Is there going to be a significant change in the way that these batteries are created, or is it just simply a matter of swapping out this new, more environmentally friendly compounds for those that already exist?
Okay, that's an interesting question. Actually, our binder use the same process for the traditional binder in terms of making the electrode material. Also, manufacturing this binder is a way easier and has a lot of catalysts that can be reused and to produce more of this binder.
I loved hearing all the validation interviews that you did. Congratulations on that. And you talked a little bit about what you learned from those in terms of the existing solutions. Can you share with us some feedback that you got in those interviews about your potential solution? What was the top things that were maybe of interest or of concern?
Actually, most of the interviews that I got, they are interested in finding more sustainable solution because the toxicity of the solvent they are using is affected the people who work with these binders. This is one thing. And also, the regulation about the PFAS material is a big issue now. They told us that like within 15 years, there will be a regulation to prevent the use of the traditional binders. So they are looking for finding a solution. Also, they mentioned that would be happy if we have a lower cost material for the manufacturing.
This sounds phenomenal. I'm curious about the manufacturing process of the binder itself. If you can maybe explain that a little bit to me and maybe what some of the challenges would be for that process.
Okay, so I'll go for technical part a little bit. So we are using a catalyst, which is iron chloride. Both are safe. And we are using ethanol mainly for this processing and water. Just mixing for a specific time of hour and leave it for the next day, dry it, and then it's ready to use. So it's pretty simple to use.
That's nice.
Thank you.
Thank you. Well, thank you.
Thank you so much for that. Our next presenter is Devin Bath, who will be presenting the project Grad Finder. Welcome, Devin.
Right. In our experience, the most difficult aspect of graduate school is not any of the work related to graduate school, but finding a supervisor for graduate school. Now, why is that? Preliminary surveys tell us that graduate students struggle because graduate students require referrals to get graduate student positions. This is mirrored. Sorry, Richard. On the other side, graduate supervisors are bombarded by emails and struggle to find graduate students because they rely on their network to distribute available positions. This is mirrored in research in general. Researchers require collaborators to maximize research funding and efficacy. The availability of potential collaborators is limited by the extent of researcher networks. So the overarching problem here for graduate students, supervisors, and researchers is that research as a whole is limited by networks. So Devin, why don't we just use social media to grow our networks? Well, if you want to scroll through goat videos, off-putting opinions, and irrelevant posts, then social media is the place for you. But if you're a busy student or researcher, you're going to need an efficient way to find opportunities within your field. Unfortunately, the resources available to you right now are limited. At GradFinder, we are developing a solution for this. We are creating a networking and position searching platform to increase the velocity of information transfer within research. But instead of telling you about it, why don't I show you? If you were to log on to our website, this is what you would see. This is your profile. You can fill in all your information for your profile here. Your searching options are here. If you were to search for an available position, you could filter by position type, research field, and location. We'll even recommend new positions for you based on your searching activity and notify you when new positions relevant to you come up. This is your news feed, positions, collaborative opportunities, and general posts such as research papers or accolades relevant to you will come up here. And we'll recommend new people for you to connect with based on your research interests and field. So to tell you a little bit about our team, we have an interdisciplinary team that has independently created 12 websites and is currently publishing research in the fields of artificial intelligence and software engineering. As we communicate with industry experts and take part in startup incubators, our team is more than capable of developing and taking to market this product. How many times as a graduate student have you struggled to find the right graduate student position? Well, how many times as a researcher have you required a specific skill set and not had that person in your network? Well, no more. At GradFinder, we are creating a vast ecosystem to revolutionize the way you as graduate students and researchers get positions, connect, and collaborate with one another. Thank you.
I think all the judges can say that was a good recovery. These things, pitches are hard. Pitches are hard. You recovered well. And if anyone else struggles throughout this, just take a deep breath and keep going. It'll be fine. It was good. Questions from the judges?
Absolutely. Kudos on keeping going. Fantastic.
Sorry about that.
I guess my question is, how would you envision making money?
Yeah, so that's actually a super, super important question. How are we going to make money off of this product? Well, what we're doing is we're buying time from people that have very, very little time. So they need a high value product and they don't need any extra clutter in this product. So what we're going to do is targeted ads. So while we're making money on targeted ads, we're also going to be giving them more opportunities through granting agencies and any kind of scholarship opportunities. So in addition to making money, we're actually improving our product by giving them more opportunities.
Thanks so much. I'm really interested, this group obviously doesn't have a lot of time, but it seems like they're going to have to be updating a lot of their information to keep it relevant, all the research, all the publications. Is there anything on the back end that's going to help them do that job as well?
Yeah, absolutely. So this is actually a pretty simple fix. This is something we integrate. We've already done work on this. You're integrating essentially your Google Scholar, right, which actually automatically updates as you go, anything like if you're using ResearchGate, so you can actually update your profile automatically as new research papers come up, and you can choose what you're actually posting to your newsfeed.
So I just wanted to touch a little bit on the advertisements. So, you know, how do you envision getting those advertisements in front of the researchers? Is this something where it's on journals? Is it coming through journals? Is it through direct mailings? Is it through events that you sponsor like this, where you can say, hey, come talk to us if you're looking for a posting? It's with, you know, just to build a little bit on the earlier question of, you know, and there's a limited amount of time, there's a little bit of amount of bandwidth by the researchers. How do you get in front of them?
Well, really, we're looking at using targeted ads actually right on the website, right? Just little banners up here or there. And then, yeah, obviously, we're going to be doing other things such as actual events. And we're looking to diversify through this as much as we can and really retain people's time on the website, because that's essentially what we're buying.
Sure. Okay, thank you.
I'll ask just one more question. Can you just share with us how far along the prototype is?
Oh, yeah, absolutely. So we've actually created most of the front end now. We're looking at integrating the back end. What you're seeing right now is just templates that are done so far. So we're getting very close. We're actually having a timeline right now of beta testing in the next 6 to 12 months. About 12 months, we're looking at talking to investors and actually getting some of that money for marketing. But up until then, right now, we're in a very, very good position. So I'd say 6 to 12 months, we're going to be beta testing.
Yeah, thank you.
Good job.
Thanks, Devin and team. We'll hear next from Ian Crook, who's presenting on the project Syphistat, a glucose-based rapid syphilis test.
Manitoba's slogan is Friendly Manitoba. But one thing Manitobans have been sharing isn't as safe as that secondhand couch from Kijiji, and that's syphilis. Syphilis cases in Canada and the US have skyrocketed in the past 2 decades. Canada has seen an 800% increase since 2011, and in the US, the CDC declared syphilis an epidemic when cases rose over 200,000 in 2022. This prompted a call for action for the swift innovation and collaboration between all STI prevention partners. Syphilis symptoms can be unnoticed, and sometimes it can be transmitted unknowingly, even from mother to infant. This congenital syphilis case can be deadly for the baby. The Canadian government recommends that over 1 million Canadians should be getting tested for syphilis 3 times per year. Do you think that's happening? Currently, the testing protocol takes 2 steps. Step 1, to test if the patient has ever had syphilis, and step 2, to see if it's an active infection. These are done in a clinical setting, as there are very few rapid tests on the market in Canada. Right now, to treat a syphilis infection for adults can cost upwards of $1,900, while a congenital syphilis case can cost $18,000. These costs and these cases this high can be avoided with early detection and treatment. Our solution is Syphistat, a portable rapid diagnostic test to detect active syphilis infection with speed and accuracy, complementing those step 1 rapid tests. It uses innovative chemistry to quantify the active infection, which is lacking from the rapid market right now. Our proprietary technology will work with a diabetes monitor, which is extremely accessible and is already in the hands of many Canadians. We estimate that this test will only take 15 minutes from pricking the patient's finger to getting a result, lowering a 6-day wait time to only 1 single visit. Using a quantity alleviates the uncertainty of binary qualitative tests that can give false positives, especially in pregnant individuals, leading to unnecessary costs and antibiotic prescriptions. Syphistat will not only lower the cost for the healthcare system and the workload, it will also increase accessibility to testing, as it can be done with minimal training for underrepresented populations and people in decentralized locations. Thank you.
Thanks, Ian. Now we'll hear some questions from the judges.
So I'd just like to ask about false positive rates. So how accurate is your test compared to the standard tests that are already being used? Is there a concern that someone might have a false positive and then not seek a follow-up at a later date?
So we are in very early stages. We are in the conceptual stages of our idea. We are 2 undergrads with an idea. We're on the hunt for a lab. But where our test shines is with the 2-step system that's required for syphilis, because it's a complex infection. What happens is patients go in for that first diagnostic test, they wait 6 days, and for underserviced populations, they might be hard to track down once you have the result. And so that's where patients are getting lost. And so
what we need as a solution here in Canada and across the world is that immediate connection between testing and treatment.
Okay, thank you.
So my understanding is that this doesn't require a new device, that this uses an existing glucose meter. That's kind of interesting. So how does that change? Like, I guess I'm interested in how the test works then, if you could maybe drill into it a little bit more detail, and how that might change your process of getting certified so you can put it in a healthcare setting.
Yeah, so the proprietary technology would turn the kind of the detection of syphilis into glucose. So the amount of glucose that you can see on the glucose monitor would directly refer to the amount of active infection that is in the patient.
Makes sense.
Okay, I have a question. You acknowledged that this is early stages, but how would you imagine going to the market?
So we believe that the fastest way to get this idea on the go is to find a lab, treat this as a master's project. We will lose some of the IP, but we hope to purchase that back. And ultimately, once we have collected data, then we could start patenting and connecting with programs that are available in Canada, such as the Can Health Network, as well as Inmari Bioinnovations, Lab to Market, to kind of help bridge that gap into commercialization and ultimately potentially selling this idea to biotech companies that specialize in manufacturing point-of-care diagnostic tests.
Thank you.
Thank you very much.
Thank you, Ian and team. We're going to take a 15-minute break. I'm not sure where we're at with the solar eclipse. And if any experts in the audience could tell us how close we are to that. But we are going to take 15 minutes, and there are glasses at the back if you want to take part in the partial solar eclipse. We will be starting the next pitch, though, promptly at 2:40. So if we could ask that you all are back by then, that would be great. Hi, everyone. We're going to get started again. And I'm hoping everybody enjoyed the break and got a glimpse of the partial eclipse. Thank you as well to the faculty for providing and organizers for providing glasses for that opportunity for some of the people who took part. Up next is Project 6. It's Shubneet Thind who will be presenting the project The Power of Electrical Current, Disease Detection in Newborns. Please welcome Shubneet to the stage.
Picture this. You just became a new parent to sweet baby Jenny just 3 weeks ago. But you've noticed that she's been weak and quiet. Concerned, you take her to the doctor where it's determined that she must undergo a muscle biopsy to be screened for rare diseases. You are devastated. And what's even worse is that she doesn't get her first muscle biopsy until she's 4 months old. She's then diagnosed with cytochrome C oxidase deficiency, or COX deficiency for short. This is a rare hereditary disorder that severely affects muscle, liver, heart, and the brain. This deficiency affects infants by the age of 2, and as seen in Jenny's case, muscle biopsies are not only invasive, but time-consuming and expensive, resulting in a delay in diagnosis. This is exactly why finding a quick, inexpensive, and early means of diagnosis is essential to minimizing symptoms and preventing long-term disability in young infants. And so, I present to you our solution, the pocket sensor for oxidase detection. This point-of-care electrochemical sensor quantifies the activity of the oxidase protein in one single cell. This point-of-care sensor works in 3 easy steps. First, you get a cell sample using a non-invasive method, like a cheek swab. And then you inject the cell sample so that it can collide with the sensor. And then With the push of a button, minimally trained personnel can get the result on a tablet, saying whether or not the sample was oxidase deficient or not. This technology will save thousands of parents the emotional anguish that's caused by traditional methods of detection, and it paves the way for rapid disease detection using sensing technology at birth. In fact, the efficiency of our method will allow all newborns to be screened for COX deficiency right at birth within seconds. And this will act as a preventative measure to rule out the possibility of having this disorder. This technology exemplifies that when biology meets electrochemistry, we can improve the quality of life for thousands of infants. Thank you.
Thanks, Shubneet. We'll now get some questions from the judges.
So you've talked about this as an issue with potential muscle concerns. Is there other types of diseases where there's a lack or an inhibition of this enzyme?
This specifically, it's called cytochrome C oxidase deficiency, is because it's that protein that's deficient.
Wonderful pitch. Can this base technology be applied to other forms of detection at different ages, for example?
Different ages, yes. There's not a limit since the cell sample can come from any individual. Other fields, yes. I think this technology serves as a basis for sensing technology in many other fields. This could be antibiotic resistance, for example.
At what stage is this technology? And then my follow-up would be, what would you do next?
Currently, the proof of concept has been established and published. The patent for this electrochemical principle has been granted. Currently, we're working on miniaturizing the sensor and generating more validating data specifically for cheek cells so that we can derive a threshold that differentiates deficient cells from non-deficient cells.
Wow. Thank you.
Thank you, Shubneet. Our next presenter is Randy Roy, presenting the project Immobilized Enzymes for Novel Flow Reactors. Please join me in welcoming Randy to the stage.
Did you know that over 50% of FDA-approved drugs come from natural products? Natural products are small organic molecules derived from microorganisms such as bacteria and fungi, and are responsible for producing life-changing therapeutics such as penicillin and lovastatin. But the challenge with natural products is in their production. Extracting them directly from microorganisms results in very low quantities. Synthetic chemistry serves as a route for making them in higher quantities. However, this often involves multiple steps, toxic solvents, and generates unwanted byproducts. We demonstrate a solution to this challenge with orsolinic acid. Orsolinic acid is a natural product primarily produced by lichen and fungi. This molecule has very promising therapeutic potential in cancer and bone regeneration, which was most recently reported in 2022. But one of the major obstacles with its use is its high price tag. It sells on the market for over $2,300 per gram. This expensive cost prevents further expansion into its research and therapeutic potential. The underlying reason for this expensive cost is reflected by how it is synthesized. The synthetic route to produce orsolinic acid starts from inexpensive orsinol in a multi-step series of reactions that utilizes toxic solvents and generates unwanted byproducts. But what if I told you that we could convert orsinol into orsolinic acid in one single step? We have been able to do this through the use of enzymes. Enzymes are powerful catalysts that enable all the necessary transformations needed for life. With our solution, we are able to use these enzymes through immobilizing them by attaching it to a solid support. This is the foundation of our technology, where we are able to bind enzymes that produce natural products like orsolinic acid. Our technology is able to produce orsolinic acid in one single step with an inexpensive precursor in water and baking soda, greatly simplifying the process with no unwanted byproducts. With our technology, we are unlocking the therapeutic potential of orsolinic acid by addressing its limited supply. But what we really want to impress here is that this technology is not exclusive to this one reaction. We are able to generate a suite of customized, immobilized enzymes that are able to access other difficult to achieve chemical transformations. This emphasizes its unlimited potential. Through immobilizing enzymes, we are creating a sustainable, less labor-intensive and economically viable process. With our solution, we are literally spinning straw into gold. Thank you.
Right on the watch. Nice job. Right on the clock. Any questions for Randy?
That was a wonderful pitch. Again, thank you so much. Curious, I love the fact that you opened up the door to a whole bunch of different areas. Where are you with the testing and how do you protect your IP around this?
Yeah, perfect. So we're currently at a TRL 4 level, so we're in early prototyping. And we are currently working with Lauren actually to do an IDF disclosure form, then we'll move on to see what capacity there is with patenting.
So for these novel enzymes, are they new to science, or are they novel? Are they enzymes that were already sort of known in science, and you've combined them for a novel methodology, or So that's basically what my question is.
These reactions are novel. We work with lichen and fungi, which have a range of many enzymes that have not even been biochemically tested or proven for the reactions. We got a whole suite of enzymes from Joint Genome Institute in California. So this is where we mean by the suite of customized immobilized enzymes that we have access to.
Okay, thank you.
Yeah.
Randy, can I throw you a curveball question?
Go ahead.
Okay.
Go ahead.
It's the teacher in me, I can't resist, okay? Could you share with us one lesson learned for that you received from preparing for today? What have you learned through the process of getting ready for today?
I think with this project and preparing for this presentation, I learned I needed to take a step back. I've gotten so into the weeds with this research that I had to figure out what matters to an audience. What is going to make people care? And I should mention as well, I'm going to learn more about this through the National Lab to Market program, where we're able to talk with stakeholders and people that are involved in this field to see where are those pain points and how my project can be innovative for them.
Thank you.
Thank you.
Any other questions? Okay, thank you.
Thank you.
Oh, this way.
Thanks, Randy. Our next presenter is Jocelyn Zabrano Alvarado, who will be presenting the project Addressing Invisible Threats, Applications of Microbial DNA to Prevent Waterborne Outbreaks. Please welcome Jocelyn to the stage.
During the Easter season of 2001, 7,000 people in North Saskatchewan were experiencing severe flu-like symptoms and infectious diarrhea, so they had to spend their holidays in the emergency room. The reason? An invisible threat, a microbe different than bacteria but still smaller than a single particle of dust, got its way into the drinking water system, making half the population sick in the city of North Battleford. Sadly, today, the same story can easily repeat because there are more than 50,000 people living in indigenous reserves in Canada that receive their water from high-risk drinking water systems. This means that they are more likely to experience a waterborne outbreak. And the largest population at risk are located in the provinces of Ontario, Saskatchewan, and guess what? Manitoba. Among the factors that could contribute to an outbreak like this is the fact that we still use water microbial tests that focus on the presence of fecal bacteria as the only indicator for water contamination. These tests have been around for more than 100 years, and they're relatively simple to use. However, they have been proven ineffective to assess water for other pathogens, such as viruses, or the microbes like in the Saskatchewan story. As I mentioned, that was not a bacteria, and it still made 7,000 people sick. So even if water is negative for fecal bacteria, it does not mean that it's safe to drink. And of course, there are other specialized labs that, upon request, could assess water for the pathogens that I'm talking about. The problem is that finding a specialized lab in remote communities is extremely difficult. So to solve this issue, don't you think that these areas would benefit from a portable device to assess water microbial quality? Something simple, like the COVID-19 rapid test, but for water. Yes, of course these areas will benefit from this. That's why we are on our way of developing this paper-based innovative solution. With this type of technology, people can assess water microbial quality on-site without the need of expensive equipment or specialized personnel, which is a common limitation in indigenous reserves. Moreover, when monitoring source water to find the DNA of pathogens of interest, we're directly preventing waterborne outbreaks in the most vulnerable regions of the country. The mass production of a device like this costs $4, way cheaper than the economic burden that waterborne pathogens cost globally, that is $5 billion. And you heard correctly, billion. With the implementation of this paper-based innovative solution, and our direct contribution to the multi-barrier approach needed to protect water and public health, we can make that these invisible microbes are no longer a threat. Thank you.
Now we'll go to the judges for any questions for you.
So you talked about detecting microbial DNA. For a device like this, would it be one sort of setup for a particular bacteria or a particular virus? How would you sort of set it up so that you're not having a lab that's testing for 200 different things or 300 different things all at once?
Awesome question. That's actually where we are right now at this project. I already went to the source of water of 3 indigenous communities, and I took water samples to find what are the most predominant pathogens in those areas. So then when we design this device, we make it as specific for the pathogens of the area to benefit the communities where we went sampling.
Thank you.
What has been the biggest challenge that you've tried to overcome while developing this innovation?
Yeah, so far, for example, we are working to try to also analyze viruses, and viruses are challenging to find and to make them work. Also, another challenge with this is to try to make this technology work with water samples or environmental samples. You have seen this type of test work with serological or with blood because they don't have inhibitors, because everything can be found in water. So developing the protocol assay to make it work for environmental samples is one of the biggest challenges that we are facing and we're trying to overcome.
Thank you, Jocelyn. Can you just confirm for me what stage you're at? This is still initial testing?
We are in the research and development stage.
Okay.
Yeah, I went sampling already. We already processed the samples. I'm processing the data, and after that will come the protocol assay development, and then the validation, and then after that the manufacturing, and then the regulatory approval.
Okay, thank you.
Thank you.
Thank you, Jocelyn. Our final presenter will be Salo Quintero dos Santos, who will be presenting the project SATS Capital. Please welcome Salo to the stage.
Hey guys, I'm going to start to actually put the pressure on you. How many of you guys have heard about Bitcoin? Everyone. Great. How many of you guys have invested on it? Just a little bit. And that's a real problem. Today, I'm actually really happy to introduce SATS Capital, an innovative rewards program that actually allows everybody that's active on the economy invest in Bitcoin without risks. Despite all the awareness about the Bitcoin and the digital economy, about only 2.8% of the people have invested in it. And despite the growth of it, all the hype, it's an open source platform that everybody could actually join, but no one is doing it. And we identified 3 main barriers. First of all, knowledge. It's pretty hard to understand the core value of it. You need to understand about economy, cryptographic fundamentals, game theory perspective. It's not easy. Aside of it, there is a technology aspect. You need to know how to keep your money safe. Now it's digital. Hackers can get it, and there is no bank that's going to protect you. You're going to be your own bank. And last but not least, money. Not everybody has the disposable income to invest in a specific asset that's have a right risk, right return proposition. So at SAT Capital, we are proud to break all those barriers with a very friendly and yet secure platform where allow our users and business to connect to that digital economy. In quite simple steps, you can actually invest in Bitcoin without any risks. With our solution, we are, for the user side, allow them to invest passively while they consume and spend the daily by day purchase. On the business side, they use it as a way to attract more customers and engage in them on their brand. And the byproducts of it is basically they're investing in their own local communities. They are investing literally every time someone else buying with them. Let me give an example. Think about students here struggling to keep their budgets away. And now they can invest in that digital economy only by on partners, while they actually are hanging out with friends, shopping for clothes. More than that, they can reference their friends and have commissions over it. Think about business that now can actually invest in their communities, create a very strong brand, being the first brand to actually give Bitcoin to their customers, while creating a community around them. So we're really proud to actually break down those barriers and bring a more actable and level playing field. We're running that company in Brazil with more than 800 users, more than 100 partners, and we're really proud to actually have more than 200% of gains over the cashback. Thank you.
Thank you. Any questions from the judges?
So you mentioned a couple of times about investing without risk. Can you dive into that a little bit more? Because I mean, there's always risks with investment. Is it at risk of not losing my money, or is it risk of making sure that we hear about stories around things that have happened around cryptocurrency lately? How is this without risks?
So the without risks here is an indirect proposition. So you would actually pay for the coffee at Tim Hortons anyway. Now you accumulate points that you can exchange for another coffee. Instead of actually get that points that accumulate for another coffee, you're gonna have little satoshis, a fraction of a Bitcoin. You would spend that money on the coffee anyway. You could show a QR code, and some of that money that's the investing market come back to you as an investment, so you have zero risk. Or you can actually buy using other rewards programs. The idea here is actually have a competitive advantage of being something that they want to have, but they don't know how to get that. Aside of it, also the technology safety. So we are taking part of it until you are actually educated enough to have your cold wallet and do it for yourself.
Great. Thank you.
So you mentioned on that last slide that you have 800 users. Could you share with us maybe the top lesson learned that you've had from a user and how you have adapted based on that feedback?
Yes. Our main challenge was not the solution itself or the value proposition, is they understand that that's an advantage for them. And then from that experience, we realized that the biggest power here is community. So the majority of the Canadians, youth Canadians that have invested in Bitcoin, they invested because a friend actually told them to do so. And we actually create an incentive inside of our platform that if you refer a friend, every single cashback that friend receives in our platform, you have a commission over it. Is within the cost for that friend to receive the cashback, we have a spread of 3% over every cashback you receive. So we split it, half stay with SATS Capital, half we distribute it across the company, the community. Users indicate, companies giving the first cashback, or a distributed licensing approach in which you like that ecosystem, you want to Bitcoinize your community, you can start to sell to the business there, and you have commissions over the people that they are bringing as well.
So if I'm the person selling the coffee, what's in it for me? What's the value for the retailer or the partner?
Usually, when you're selling your products or service, you have a margin over your product or service that you use for marketing purpose. Instead of spending it on Google or Facebook, that goes to Mark Zuckerberg and Sergey Brin, invest in your local community. Over time, it might pick up, and then more and more people are showing QR codes for you, or not, and then you don't have anything to lose.